Welcome to Jerry's Gene Scene. Today I'm going to talk about linked genes. But before we do, let's take a look at some of the previous videos on this playlist. We initially learned about the monohybrid cross, where we looked at the eyes of the phantasma. We had the yellow eyes and the white eyes, and we learned about dominance and recessive genes, and we learned about the 3-to-1 ratio that results. Then in the next video, we learned how to tell the difference between a homozygous and a heterozygous for the dominant trait when the phenotypes both look the same. Recall from that a very crucial ratio is the 1-to-1 ratio of the test cross. Then we learned about the law of independent assortment, and if we are looking at two different traits at the same time, it's called a dihybrid cross. In this case, we looked at the eyes, and we also looked at the number of legs. And when we did that, we found the 9:3:3:1 ratio. Now, if you do a test cross on that dihybrid cross, you would find a 1-to-1 to 1-to-1 ratio when we talk about dominant-dominant, dominant-recessive, recessive-dominant, and recessive-recessive as the way that we look at the phenotypes of the offspring. Let's go back to our props just one more time. If we have the yellow eyes and we have the number of legs, we can see that these can be sorted independently of each other. But we found on our phantasma a new trait, and that is the trait of what do the antennae look like. Now, we could assort the antenna separately from the legs, but the antenna and the eyes are apparently attached together. Let's see what happens when we call these linked genes. So we discovered two new phenotypes of the phantasma. We've got the white eyes, and it's got feathered antenna, and we've got the yellow eyes, and it has a straight antenna. So let's check this out and see what happens when we cross these. So with the white eyes and the feathered antenna, we end up with gametes that are only white eyes with feathered antenna. And when we look at the gametes of the yellow-eyed and straight antenna, we end up with the same thing. But when we cross these, notice that we get yellow eyes and we get feathered antenna. And that seems to be in 100% of the cases. From what we learned previously then, we would say that yellow eyes were dominant and that feathered antenna were dominant. Furthermore, we would predict, after looking at the eyes and the number of legs, that we might end up with a 9:3:3:1 ratio when we cross these guys. However, when we form the gametes from these F1 cross with the eyes and the antenna, we discover that we have the eyes and the antenna seem to stay together in both. So we'll form a little Punnett square here, and we'll occupy it in a 2x2 format, as there's only two types of gametes that are possible, yellow-eyed and straight antenna, or white-eyed and feathered antenna. So let's now populate the squares. We can see in this case that the eyes are in a 3-to-1 ratio, just as we predicted from the monohybrid cross. And we can see that the feathered antenna are also in a 3-to-1 ratio, just as we predicted in the monohybrid cross. But because these are linked genes, we also end up with a 1-to-2-to-1 ratio of the overall phenotypes that are there. Now, we would have had different results had the dominant alleles been both on the same gamete, as opposed to where we had one that was dominant and one that was recessive. But we'll use that information to map the genes. Linked genes played a very important role in the history of the mapping of the chromosomes long before any genome projects were underway. In the next video in this series, I'll take a look at how that mapping was done. You can subscribe to my channel by hitting the button on the left, and you can go back and review all of the videos in this playlist by clicking the link on the right. I'm Jerry Ward at Genome Alberta. Thanks for watching.